My name is Willie. I'm one of the professors in the department, and I'll be introducing you to all your wonderful speakers. So before we start, tonight is an evening of celebration. We are celebrating two things. First of all, we are celebrating the youngsters, the undergraduates who are now completed with their studies. They are now chemical engineers. And secondly, we are celebrating the postgrads of this department. The postgrads really do all the research work in our department, and that's not the only thing. They're very involved with the undergraduate teaching, you know, whether it's by formal teaching as being a teaching assistant, or whether it's being through the group, through the projects the fourth years do. So we want to give a big hand to the postgrads. So they keep telling me that this group of chemical engineers from their third year on is the most intelligent group of students you'll get anywhere in South Africa. Okay. I am, however, a scientist, okay, and you know, I like to measure these things. So I got this very special app on my phone. It's only available to professors, and what it does, it measures the average IQ using the Wi-Fi network, okay. So I can do a quick brain scan whenever I walk into a venue. Okay, so I do this quite often. I walk around the university, just hop into a lecture hall, take a reading, you know, and I take a lot of readings. Of course, I record my data. And I'll tell you, when the chemical engineering class is sitting there and I take the reading, it's the highest you can get. Okay. Only once had I had it higher, and that's when I bring all the postgrads together. Okay, we even distill them a bit further. Okay, so it's an honor to work with such intelligent people. Okay, but intelligence is something that's a gift, but it comes with responsibility, because the intelligent people are the people who will change this world and this country. Okay, so we as lecturers often ask ourselves, are we enabling this intellectual force that we sit with for 4 years? You know, are we enabling enough so that they can change the world? Unfortunately, there's not an app available to do that. Okay, but I'll tell you one thing. I just did a scan, okay, and the results are in. I'm not allowed to reveal numbers, but the number is in front of me, and I can tell you, surprisingly, it is exactly the same as the chemical engineering class. You know, what does this mean? You know, is it because we have so many postgrads here? Or maybe, you know, how can the parents be so good? You know, maybe there's a genetic link. Okay, so enough about measurements. Let me introduce you to someone who measures and then try to control. The head of our department, Philip DeFaul. It's all yours.
Right. Good evening to everyone. If you read on one of the first pages in the brochure or the booklet that we handed out, I have my speech written out so that I don't make any terrible mistakes in front of such a wonderful audience. So I'm going to highlight a few items. The first one, of course, is to welcome all. And this year is a special year because this is the first time that we have this event to include not only our final year undergrads, but also our postgrads. So Willie made very sure that we don't forget about that. I have to mention it was his initiative. So congratulations, Willie. I think it was a perfect idea, and I'm sure that later on you're going to agree that that is the same. But we are a numbers sort of orientated group of people. And for that reason, I've been keeping track about numbers. So we have 81 final year students in this audience. There are going to be, if I'm not mistaken, 81 graduates, more than the ones that we already have. But quite a few things. Many a slip between the cup and the lip is something that I remember from my vague past. We started many years ago. This department started in 1959, and the first group of students who registered for chemical engineering actually registered for mechanical engineering. And when they were halfway through, then the Department of Chemical Engineering was established and they graduated as chemical engineers. In that particular year, I think there were sort of 7 graduates. So we've grown because numbers have exploded really. I just want to highlight a few interesting items that we all got to learn over the past number of years. The first one is fake news. And since we are very critical about everything that we do, we also have to live with this concept of fake news as we all do. I can assure you nothing that Willie has said is fake news. He does have this little monitor and he does measure the IQ of people. How he does it, we will look into that later on. Next thing is we are inundated with terminology that we don't really know. I highlighted them. I spoke about I mentioned the 4th Industrial Revolution. And last weekend when I was driving to work on some radio program, they said that the 5th Industrial Revolution started in 1999. So the 4th Industrial Revolution is long gone. The Internet of Things, wonderful buzzword, but it gets worse. I listened to a TV program one night and one of these scholars in a different category of not science at all, they talk about the difference between people being exploited and people who become irrelevant. And they are warning us that we are, as human beings, are becoming irrelevant. So it's up to us to decide whether we agree with that or not. So are we ever going to stop with this? And we don't want to. Bottom line is, we want to produce self-thinking individuals who will not take anything for granted and who will fill the world with being critical and being productive. So I want to end this by looking at a phrase that is often misused. And this is how it came through. Someone said, I'm going to send you into the world to preach the gospel. And you can regard gospel as whatever you wish, but to preach the gospel. And if you have to, use words. So if I apply to our group of young graduates who are going to enter the real world very soon, I want to tell you, practice chemical engineering, don't only preach it. And with that, I hope that you're going to enjoy this evening and that you are going to stay with us until the bitter end, and that's it. So thank you very much for being here and enjoy the evening.
Ladies and gentlemen, posted you see outside are products from the course, the research project as they call here, and there's 3 prizes going to be awarded for students in the research project. Later on it's going to be the best project, the best poster, but we already know who the best oral presentation is, and this gives me a pleasure to introduce the Sekho, Ophelia Siabi, prefers to be called Sekho, and she's going to talk about the undergraduate.
Good evening, ladies and gentlemen. My name is Lesego Siabi, and today I will be speaking about the effects of different environmental factors on the degradation of butyric acid by a co-culture of indigenous butyric acid degrading bacteria. I have as a surrogate to pitlitrin odor control. I have been working on this project for the most part of this year under the alongside Mr. John Njalamano and under the guidance of Professor Evans Chira in the Water Utilization and Environmental Engineering Unit. To get into a brief motivation for the study, former Secretary General of the UN, Mr. Kofi Annan, once alluded to the fact that the fight against infectious diseases can never be won before we have tackled the battle for safe drinking water, sanitation, and basic health care. The word sanitation is highlighted here, not only for the fact that this is the main focus of the study, but also because it is known that it is known that the provision of adequate sanitation in communities indirectly caters for the other two problems listed here, as the containment of human excreta interrupts the transmission of disease-causing pathogens that pollute water and adversely affect domestic hygiene. On Monday this week, the United Nations observed their annual World Toilet Day, and it was established that 4.5 billion people, that is more than half of the world's population, does not have access to what is termed safely managed sanitation. According to the World Health Organization and UNICEF, 12% of the world's population practice open defecation. That is a whole 892 million people having to excrete feces in the open public because of a myriad of reasons, including traditional beliefs, lack of access to sanitation facilities, or access to lavatories that are of such poor standard that people see it as a better option to defecate in the public. Stats SA also tells us that 4% of South African households are faced with this predicament. To solve these problems and improve overall standard of living for people in low-income countries, simple improved sanitation methods have been developed, and today we see that 1.77 billion people make use of pit latrines, or as it is known by its colloquial name, the long drop. To achieve Sustainable Development Goal 6.2 around water and sanitation, it has been found that pit latrines provide an adequate solution to the problem of open defecation for low-income countries. The main reasons for this is around their affordability, their easy installation, and their effectiveness in containing human excreta. The reality in developing countries is that the number of pit latrines that are used is set to increase because of the pros that are linked to their use. Nevertheless, these facilities are not without their limitations. One being the noxious smells that are emitted from a buildup of fecal sludge. It was found that the fatty acid butyric acid is one of the predominant odor-causing compounds detected in pit latrine emissions. In its pure state, this chemical has a very pungent smell of sweet rancid that brings about a lot of discomfort to the senses. So how do we solve this problem? Where does the engineering fit into all of this? In order to make pit latrines smell better, we have to get rid of the cause of the problem, that being the chemical butyric acid. Bioremediation is the use of microorganisms to transform hazardous compounds and convert them to innocuous products such as carbon dioxide and water. The Department of Water Utilization was able to identify 9 bacterial strains that are known to completely and effectively degrade butyric acid within 24 hours from concentrations as high as 13,000 parts per million. It should be noted that humans are able to detect butyric acid from concentrations as low as 10 parts per million. Thus, at 13,000 ppm, you can get a feel for how tormenting it can be to be inside one of these facilities. As much as these strains effectively degrade butyric acid, in order to produce an effective odor control product, their degradation rate needs to be optimized. The aim of this investigation was thus to evaluate the butyric acid degradation efficiencies of all bacterial strains to form a bacterial consortium that elucidates the cooperative action of different bacterial species in the degradation of butyric acid. After doing this, the effects of different environmental factors on the degradation of the super consortium were assessed to establish the basis for identifying the optimum environmental conditions for the degradation of butyric acid. Now, how was all of this achieved? All experiments to test the degradation of butyric acid were performed at the conditions shown on the slide above. 1,000 parts per million of butyric acid was added as the initial concentration to each reaction solution. All experiments were run for a 24-hour period, and aseptic samples were taken every 4 hours. Each of the biotic reactions were assessed against an abiotic control reaction. This was done to cater for any losses of butyric acid that occur as a result of photodegradation or evaporation. The percentage degradation was then determined using the equation that is shown on the screen above, with RD representing the percentage degradation, AC representing the butyric acid concentration of the abiotic reaction medium, and AS representing the butyric acid concentration of the biotic reaction medium. The bacterial consortia were investigated using the top 6 performing bacterial species. 19 bacterial consortia were indigenously constructed by applying combinatory statistical mathematics using the formula shown on the slide above, where N represents the total number of bacterial strains and K represents the number of bacterial strains in the consortium. A Python program was then written to compute the bacterial consortia. This Python program took in an object set of the bacterial strains and a K value and produced the associated bacterial consortia. The environmental conditions that were investigated were the temperature, pH, inoculum, and the inoculum size added to the reaction medium. When one independent variable was changed, all other variables were kept constant at the conditions that were shown on the slide above. This procedure was followed because of the time that it was allowed for the project to take place. A full factorial experimental parameter analysis would have been a more feasible procedure to follow to investigate the interactions of these parameters and how they affect the overall degradation of butyric acid. The effects of these environmental parameters were tested for their performance by measuring the degradation of butyric acid. So what did we find out? The figure above shows the residual butyric acid for each of the 9 bacterial strains at 4-hour time intervals for a 24-hour reaction time. I will briefly explain the legend that is used here. Here, what we did is we abbreviated each bacterial species according to its genus and its species name, where AS represents alkaligenes strain SY1, AA represents Achromobacter anemicus, PA represents Pseudomonas aeruginosa, SM represents Serratia marcescens, AX represents Achromobacter xylosoxidans, BC represents Bacillus cereus, LF represents Lysine-Bacillus fusiformis, BM represents Bacillus methylotrophicus, and BS represents Bacillus subtilis. From this graph, we can see that the species AS and BS were able to degrade butyric acid within 16 hours. The species LF, PA, SM, BM, and BC were all able to degrade butyric acid within 20 hours, while the other 3 bacterial species showed very slow degradation and were only able to degrade butyric acid after the full 24-hour reaction period. From the results, it can be concluded that the top performing bacterial species in order of decreasing performance are Achromobacter xylosoxidans, Bacillus subtilis, Lysine-Bacillus fusiformis, Bacillus cereus, Pseudomonas aeruginosa, and Bacillus methylotrophicus. As mentioned before, the Bacterial Consortia was constructed using the top performing bacterial strains by means of mathematical combinations. A total number of 19 bacterial consortia was constructed, and each of these consortia were tested for their butyric acid degradation efficiencies. The graph above shows the performance of all 19 bacterial consortia. From this graph, we see that the consortia C1, 2, 3, and 7 were all able to degrade butyric acid within 16 hours. And from further investigation of the results, it was found that consortium C3 was able to showed the best degradation rate of butyric acid. It must be noted that this consortium was made up of Lysine-Bacillus fusiformis and Bacillus subtilis. It must be noted also that the top performing bacterial strains, namely Achromobacter xylosoxidans, is not present in this bacterial consortium. At 16 hours, the bacterial consortia containing different bacteria showed varying performances. The reason for this could lie in the metabolic and physiological inadequacies of the bacterial strain in the consortium that were compensated for by the presence of other bacterial strains with appropriate complementary physiology. Or it could be that one bacterial strain was able to take up the intermediate metabolic processes of the other bacterial strains that could have been toxic to it, and thus affected its own metabolic processes and slowed down its own degradation of butyric acid. It is assumed that this is what happened with Achromobacter xylosoxidans. To ascertain this conjecture, a full analysis of the specific interactions between the bacterial strains would have to be analyzed. Measuring the effects of temperature on the butyric acid degradation of consortium C3 showed that the highest butyric acid degradation rate was achieved between a temperature range of 25 to 35 degrees Celsius, with the optimum temperature being at 30 degrees Celsius. In microbiology, it is known that the temperature has a great effect on the enzymatically catalyzed reactions and influences the diffusion rate of the substrate compounds into the cell. At lower temperatures, the catalytic capacity of the enzymes is reduced dramatically, while at very high temperatures, the enzymes are completely denatured, which thus affects the activity. pH is another important parameter for microorganisms. The results show that the degradation of butyric acid was optimal between a pH range of 6 and 8. This suggests that the butyric acid degrading enzymes have their optimal activity in neutral surroundings. It is worth noting that the bacterial strains may have mechanisms to modify the pH of their surroundings. It was found that over time, the pH of each of the reactions tended towards a neutral pH of 7, even outside the pH range, which is optimal for these bacterial strains. For both bacterial species, the butyric acid showed a positive correlation with the inoculum size. In the study, the optimum degradation was found at an inoculum size of 2.5. It is worth noting, though, that due to the limitations of the equipment that was used, the highest optical density that could have been achieved was an optical density of 2.5. Thus, we could not test higher optical density. It is known from the literature that an increase in the cell density in a reaction increases the rate of the substrate consumption. However, at very high cell densities, when the cell densities become too high, competition between the bacterial strains exists, and thus a survival of the fittest phenomenon takes over. To conclude, the study showed that the degradation of butyric acid can be improved by using a co-culture of bacterial species as opposed to one bacterial strain. The presence of bacterial consortia also resonates with the intrinsic environment of a normal pit latrine. It was found that the bacterial strains Lysine-Bacillus fusiformis and Bacillus subtilis together in a consortium, showed the best performing butyric acid degradation. The optimum temperature range for the butyric acid degradation was a temperature range between 25 and 35 degrees Celsius, the optimum pH being between 6 and 8, and the optimum inoculum size was found at 2.5. To conclude, I would like to thank Professor Evans Chira and John Njalamano for their continuous guidance and support throughout this project, and to thank all the students and staff in the Water Utilization Unit for their consistent support and making the working environment as feasible as possible. Thank you.
So what we have here today, we're going to have 3 presentations from postgrads and from different groups in our department. We had an internal competition where we had numerous postgrads presenting their work, and they decided among themselves who the best presentations are to present work that we're doing in our department of research. And I'm glad to introduce you to Bianca Geerfers, who's our first speaker, and she'll be talking about, well, you got the topic.
Thank you, Billy. My presentation today will be slightly different in as that you will see terribly few results, and my aim in this is to motivate you in this field and explain to you what I'm working in. So we are going to talk today about water, and more specifically about renewable energy generation. So my name is Bianca Geerfers, and I work in the group of layer double hydroxides under the supervision of Prof. Laguscachny. I'm going to introduce you today to the topic of water splitting using hydrogen as a renewable energy generation technique, and I'm going to look at this with you using the process of photocatalysis and using the material layer double hydroxides. So before we start this off formally, I would like to make each of you aware of the fact that a lot of you, by wearing jewelry tonight, you are actually walking around with materials that are quite commonly found in water splitting catalysts or catalysts in general. So some of you might be wearing platinum, gold, silver, copper, or nickel, and these we can actually find in the catalysts. Now, why this is really important is that all of you, I'm fairly certain, know that none of you have generated hydrogen by spontaneously generated hydrogen by putting your hands underwater when washing your hands in normal daylight. So in order to discover why this is the case and how we can generate hydrogen from these materials and what materials we actually need, I'm going to go through the photocatalytic process with you and introduce you to catalysis. So I want to introduce you to the water droplet and the water molecule. Now I'm fairly certain that all of you will agree with me that when we shine light on this water molecule, we will in fact not see it spontaneously decompose. We will also not see that when we put a catalyst into the water and shine light on it, that we form hydrogen and oxygen from this. Now the reason for this is that water splitting actually requires quite a lot of energy. 16 megajoule per kilogram, in fact, which is approximately equal to boiling 47 kilograms of water, so this is quite sufficient and quite a lot. Now what happens though when we submerge our hands in water? Why do we not generate hydrogen? So the first thing that happens is surface adsorption. This is one of the fundamental principles of catalysis and is definitely required. So a water molecule is polar. It consists of 2 electron-deficient hydrogen atoms bonded to an oxygen atom, and the oxygen atom has 2 lone pairs of electrons depicted here by the head on the slide. Now if I bring this water molecule into contact with a catalytic surface that is electron-deficient, I can observe something called surface adsorption. Now usually catalysts are metals. If we, for example, choose calcium or lithium, however, we will see that this spontaneously reacts and we do form hydrogen. Now the reason for this is that when we throw metals like this into the water, we form a hydroxide layer on the outside and we form hydrogen in the process. Unfortunately though, this is not a catalytic process, this is a reactive process, and once we have covered the surface in hydroxides, we will no longer generate hydrogen. So we need something that can generate hydrogen on a more sustainable way. So how we do this is that we choose catalysts, for example, transition metals, or generally catalysts that are electron deficient, but not as electron deficient as metals, for example, that can spontaneously react with the water. So once this process is complete, we will have adsorbed a layer of water onto the catalyst. However, we will unfortunately still not have split water. So what do we need? Unfortunately, the process that we need in order to split water at atmospheric conditions is in fact not catalysis, but electrocatalysis. So what this implies is that we actually need an electron or a charged surface with electrons on the surface. Now, if we repeat this process, so we have a water molecule approaching the surface, the water molecule will rearrange such that the electron-deficient hydrogen atom is closer to the charged surface. The hydrogen atom will then realize that it can obtain an electron from the surface, and it will split off the hydroxy group, which leaves negatively charged. If we repeat this process and end up with another hydrogen atom adsorbed onto the surface next to this one, they can bond together and we have hydrogen evolution. This is one of the possible mechanisms for hydrogen evolution. There are very many more, or some more, depending on what conditions you use. As engineers, however, as you have all graduated, you've all gone through CRO, you all know that having catalysts, that have active sites, any catalyst needs active sites, but having a catalyst made purely out of one material is really quite unsustainable. We can't fill our reactors with kilograms of platinum, for example. Who's going to pay this? So what engineers and scientists have come up with to solve this problem is that we use a support material, so a support material like this, a flat surface. If we do this really well, we will increase the surface area of this material. This could, for example, also work with our platinum. It's just very expensive. And then once we have achieved this, we then dope or somehow modify the material to have active sites on our material, which then can take part in our catalysis process. So what the catalyst gives us is a stage, so to say, for our catalysis with the morphology of the catalyst, and it also provides us with the active sites on the catalyst. And by varying these two compositions or these two properties, we can obtain unique catalytic materials. So now we have covered some of electrocatalysis, a very tiny part, and some of catalysis, also a very tiny part, but I told you that we were going to look at photocatalysis. So where does the photo part come in? Well, as you might have noticed earlier, is that we need a charged surface. Now, if you work in electrocatalysis and you form hydrogen by electrolysis, which is the process of submersing a cathode and an anode into water connected by some sort of voltage or a battery, for example, then you can generate hydrogen in this manner. Unfortunately, however, if you work with a powder like I do, you can't just go and connect the powder to a battery. So we need to be a bit more clever about this on how we are going to get an electron to the surface, and this is where the photo part comes in. So in photocatalysis, we use the fundamental property of light, which is that it gives us photons, and these photons all carry energy, and the energy is represented by the wavelength of the photon. Now on Earth we are lucky because we have solar light, and the solar light is represented by the solar irradiance spectrum. Now on your left-hand side you will see the UV side, which is very highly energetic, but we get very little of that. You will see the visible light range, and you will see the IR light range, which is quite low in energy. For the person that has now listened to my talk quite well and has grasped the thing that we need quite a lot of electrons in order to get our catalysis process going, you would really want to harvest as many photons as possible, because we are going to generate, and I'm specifically putting this into quotation marks, generate electrons from them. So I would like you to please keep in mind that in catalysis or in photocatalysis, we would really like to use all of the solar spectrum. Before we go to this part though, I want to discuss with you what material properties we need. So I'm going to introduce you to three material classes. The first being metals, then semiconductors and insulators. Now these three material classes have, for example, one thing in common, and that is that they all have a conduction and a valence band. Now what are these? A valence band should ring a couple of bells from chemistry. This is where we have our electrons that are capable of performing reactions swirling around in our electron cloud. So the conduction band, and this is the nice thing about metals, if we manage to get a metal into the conduction band, we can actually conduct electricity. So in a metal, the conduction and the valence band are so close together / are overlapping that the electrons can easily move from the valence band into the conduction band, and thus we can conduct electricity. In the semiconductor and in the insulator's case, we have a gap between these two bands, a band gap. Now, in order for us to move an electron from the valence band into the conduction band, we need to supply energy, and this is where our photons come into play. So by supplying a photon to this material, we can actually, if the photon has enough energy, motivate this electron to move from the valence band into the conduction band, and at that point we can then conduct the electron away and use it for our reactions at the catalytic surface. In an insulator, this gap is so large that you would have to supply incredible amounts of energy in order to get the electron to move from the valence band to the conduction band. In the semiconductor, we are lucky enough that we can move with quite little energy, and what is supplied to us by the solar irradiance spectrum, move an electron to the conduction band by making it absorb a photon. So let us look now at the photocatalytic process. What does it actually require to have a catalytic reaction at the surface of such a particle? I'm going to look at this with you with semiconductor particles, because that is what is most frequently used. You will know this term also from photovoltaics, for example, but they can also react as act as catalysts. Now, if we have light that hits a semiconductor particle, we can be able to separate the charges in the semiconductor particle. How we would achieve this is by supplying energy with a photon, and this is where you should please remember back to the solar irradiance spectrum, and that we would want to use all of it. So if we are able to use all of the solar irradiance spectrum, that somehow needs to be linked to the material, right? So how this is linked is that if the band gap of the material is small, then we can use light of lower frequency or of higher frequency, IR spectrum, so lower energies, in order to give our electron energy to go to the conduction band. Now, if we have a large band gap, we would need a lot of energy, so we can only use light from the UV range. And this is why when you hear catalysts or look at PV cells, for example, they are very frequently used in the UV range, because it is just so much easier to have higher energy input into this. There is also a second property, but we're going to look at that in a second. Let us assume for a moment now, though, that we have managed to generate an electron and a hole. So what happens now is that we transfer the charges from this separation site to the catalytic surface, where they can then take part in oxidation and reduction reactions. Unfortunately, however, this process is not quite so ideal, so we will have some surface recombinations as well as bulk recombinations, and this really makes our quantum efficiency and quantum yield of our material skyrocket, or, well, downside in the other direction, so to say, go right down into the cellar if we have these properties incorrect. Now, the other thing that you might have noticed is the little blue thing of a voltage appearing there. So what does that have to do with this whole process? The process of photocatalysis is actually also photoelectrocatalysis. So we still need that electric potential difference in order to perform our reaction. Now, how this plays into our material properties is that we would need a band gap of at least 1.23 electron volt in order for us to generate electrons and holes with enough energy to, when they get to the surface, take part in this reaction. And this is why we cannot use all of the solar spectrum so easily. Because you need 1.23 volts at the least, you have to get a material which has more than that in terms of band gap, in order to account for example, losses like the recombinations or other quantum losses that are involved in this process. Okay, so now that we have kind of understood what the photocatalytic process is all about, you will realize that in order for a photocatalyst to work, you really need quite a couple of stars to align. You need a catalytic surface, a morphology for your material. You need active sites with this material. You need a semiconductor particle that has the ability to absorb light, but also have the band gap of the material positioned across the correct oxidation and reduction potential. You need an electron and a hole which have made it to the surface, and you need, at this point, the electron and the hole interact with the water to take part in the oxidation and reduction reactions. So now the big question, where does my project fit in here, and why am I telling you this? So what I did in this process is that I work with layered double hydroxides. Now, layered double hydroxides are really quite nice materials because they can act as semiconductors if we construct them correctly. They have this layered structure there with the layer, interlayer, layer, and they have the ability to be modified in terms of their metal composition, divalent and trivalent metal ions, as well as the interlayer with anions and water. Now, what I did in this is that I decided to modify the crystal lattice structure by including a variety of different metals into my LDH. I then characterized the material with XRD, XRF, SEM, it's quite a list, EDX, TGA, and UV-Vis to get some of my material properties, but also to make sure that I actually have the material that I wanted to work with. What I wanted to achieve at this point is that I wanted to generate a material which has a high surface area. I wanted to have a material that has increased light absorption. I wanted to have a material with a changed number of active sites and where I can change the composition. And my hypothesis in all of this was that by modifying the LDH's composition, I can modify the band gap. Now let's look at some results. So the morphological changes are the first thing. We started off with a very low surface area material. That's the material we usually work with in the group. It's quite crystalline, it's very well defined. It has a surface area of approximately 5 square meters per gram, which is really not so high. It's also a micro material. I then went on and modified this material to be a nanomaterial with a high surface area of 50 square meters per gram, and then further on changed the porosity of the material to obtain a material with 60 square meters per gram. Now here is one of the series that I modified to get from the 50 square meters per gram all the way to the last one, and you will see that the porosity changes quite significantly. But why am I interested in changing the surface area? That one we've answered at the beginning, because it can increase our activity, but it can also do something different for me, and this is why I also wanted to change the morphology. And that is that my idea behind this was that when I have a photon hitting my catalytic or my crystal platelet in this case, I would really like to funnel it down and not reflect it back to the environment. So I would like to have it hit all of the catalytic surfaces until all the light is completely absorbed. Now we should be able to see this, and luckily for me we do. Here is a scan of the absorption. Oh, this one too far, sorry. One of the absorptions, so you can see here how we managed to increase the absorption of the material. And one of the nice side effects that you can also see here, well, not quite side effect, it was wished that way, but we also increased the light absorption from the UV light only, the black figure at the black line at the bottom, all the way to reach into the near IR region. So if you modify the composition, you can get even funkier results. These are some of those where you can see here I calculated the band gap for some of them, where we moved from a very wide band gap material 3.23 electron volt all the way to the visible light material 1.73 electron volt over there. And what is also quite nice is that we managed to get multi-electronic structures in these materials, as well as materials with multiple band gaps. I'm fairly certain, though, that if I ended this presentation here at this point, none of you would have been very satisfied because you haven't seen any hydrogen evolve yet. So the way we test for hydrogen is actually quite a simple setup. We need a light source, we need water, we need a catalyst, and we need something to analyze our gas. I use GCMS for the gas analysis, and I use a solar sunlight simulator. Unfortunately, the gas analysis system was at the point not calibrated, so I don't actually have rates, so I'm sorry for that. The only thing I can provide at this stage is results that we have actually managed to make hydrogen from this. And these look like that. Now, you see here small little peaks, and these might not look like a lot to you, but it will become a bit clearer to you why these are so important for me. And the reason that these are so important for me is that when you look at literature, for example, you will find that the water splitting using layered double hydroxides is very infrequently investigated. Up to this point in time, I found approximately 20 papers investigating this across the last number of years, which is quite surprising, seeing that it is such a developing field. And what is even more surprising is that all of these research groups have used sacrificial agents in order to generate the hydrogen. Now, what a sacrificial agent does is that it simplifies the reaction in that it provides an alternative pathway, and it also provides you with an electron or hole donor, so our catalytic reaction can progress quite a lot easier, and we don't specifically need the band gap to sit exactly across the water oxidation potential, water reduction oxidation potential, but across the whatever reaction we choose to do, either hydrogen evolution or oxygen evolution, and the respective sacrificial agent that we use. So why I liked my previous results much more than the ones I'm going to show you now, oops, no, sorry, one too far. These are the results that are generated when using methanol. Now, you might think that these are a lot more impressive. I mean, we can see really big peaks, and this looks really amazing. The problem, though, with this is, and that is why I'm much more happy about the other results than these results, is that the previous results that I showed you actually indicated that I was able to simultaneously, on one catalyst, generate hydrogen and oxygen. This is quite a feat in itself, because you all know that hydrogen and oxygen react, so that I got something out is nice, but it is also great because this is not frequently achieved. I haven't found a paper yet that actually investigates this. The best thing that I found was a group that used a sacrificial agent to do both the evolution and the evolution of oxygen and hydrogen using two different sacrificial agents. So this is really why this result is super exciting. What does it mean? Can we stop here? Definitely not. We need to repeat these results. We need to make these results be a bit more representative. We need to test more materials. But what it does mean is that with the background of this, having managed to generate a material that can do both of these reactions at once, we can move forward into the future, being able to improve our catalyst and hopefully at some stage get something that actually does a proper hydrogen evolution. Thank you very much.
You'll be seeing that there's a lot of Latin names being thrown around. Latin names is what we use for speech. Really, what we need to do as chemical engineers is to learn from nature to solve the problems that we have inflicted on nature. You've got another Latin name coming up. I'm presenting to you Monique Geyer. She's an expert on the fungus Aspergillus oryzae.
Good evening, ladies and gentlemen. As you've heard, my name is Monique Geyer. So before I introduce the title of my project and I see that glossy look in your eyes, you know, what am I talking about, and you're thinking about what you're going to do this weekend, I'd rather ask you some questions. So who here would like to earn a salary of, say, 30,000 Rand a month? Nobody? Okay. Who would like to retire while earning that salary? Okay, one or two people, interesting. Okay, so I don't actually know how to do that. If I did, I wouldn't be here, and you haven't been lulled into another Amway presentation. I'm just trying to get your attention so I can introduce my title, Malic Acid Production by Aspergillus oryzae, the role of calcium carbonate. So the really three important terms in this is malic acid, Aspergillus oryzae, and calcium carbonate. So I want you guys to concentrate on those three as we go through it. So the traditional petrochemical, what we all associate with chemical engineering is petrochemicals. We get fossil fuels, we make petrol from it, we make other products from it. But now I have begrudgingly accepted this title of millennial, and I want to turn that idea of chemical engineering upside down and talk to you about biochemical engineering. So the way this works is we use renewable resources to get bio-based products that are more sustainable towards the environment. So how would we get this? We would use biomass. So biomass is readily available. And we just let this biomass decompose, we get some sugars, and now we can use these sugars to produce a variety of chemicals that we would generally get from petroleum. So what are these chemicals? Well, we get three main dicarboxylic acids that are researched in our group. That's fumaric acid, succinic acid, and then the one I'm researching, which is malic acid. So This is a really fancy structure there of malic acid. I know it doesn't mean much, but malic acid is a C4 dicarboxylic acid. Currently, the demand for malic acid is 200,000 tons per year, but we are only producing 60,000 tons per year. That means we've got a market that we can push into to make more malic acid. Another important thing about malic acid is that it has a D isomer and an L isomer. So what does this word mean, isomer? So you can think of it as atoms, as these Lego blocks. So I can build different structures using the same set of Lego blocks. So if I look at the structure I built, I can build a plane or I can build a train, but they consist of the same set of Lego blocks. What does this mean? Why is this important? Well, this is important because our malic acid is actually the natural form of malic acids. That's the one that we get in fruits, such as apples and watermelon, and it's also naturally produced by a variety of microorganisms. So why malic acid? What can we use malic acid for? So it's used in metal cleaning and finishing, and it's also used in electrolysis plating. We use it in agriculture to control the soil pH. We can make polymalic acid, which is a polymer, a biopolymer. It's also used in a variety of cosmetics. We use it in pharmaceuticals. If you go to Discam, you get that really big aisle. It has, calcium malate and all of those different ones that make your bones nice and strong. And then we also use it in textile finishing, so for fabric dyes. But then the most important one is as an acidity regulator in the food and beverage industry. So what this means is that, those gummy sweets that you like to buy, it has that really nice sour taste, or, that salt and vinegar chips that you like, or those soft drinks, they all contain malic acid. They all use malic acid to give you that really nice tart taste. So that's why it's really important for us to get our malic acid. Since that's the natural form that's in fruits, we want to digest that in our food as well. So how do we then make malic acid? So going back to the traditional petrochemical synthesis of most chemicals, we would use fossil fuels to make, for example, malic anhydride, which is a chemical that's used in a variety of it's used as a starting chemical for a variety of other chemicals. Then we would use this maleic anhydride and convert it to, say, fumaric acid and maleic acid, and then we would use catalytic hydration to then make malic acid. And then whatever else we make, we just give to the birds. That's their problem. So now what's the problem with chemical synthesis? We've been doing it for so many years. Why now change? So this process uses really high temperatures and pressures, making it economically unfeasible, and it also it produces a racemate mixture. So what this means is I get L-malic acid and I get D-malic acid. So now I've got a problem. So since I want to make sour worms with this, I can't give you D-malic acid in there now, and it's really hard to separate. So now we need to find something else. So what's the next thing we can use? Well, now we can use enzymes. So enzymes are proteins that facilitate the conversion from one thing to another thing. So this enzyme, for example, is called fumarase. So there's 4 of them. So what fumarase does, it actually takes fumaric acid and it converts it into L-malic acid. So enzymes are naturally produced by a variety of organisms, and we can easily mobilize this in a big reactor. We can put it into a membrane, or we can just use immobilized cells in the forms of microorganisms that actually produce this fumarase and then convert it. So this has higher productivity, meaning that we can make more malic acid quicker. It has higher stereoselectivity. So what that really big word means, it doesn't mean like I'm going now to Hi-Fi Corporation and being selective about what stereo I'm going to buy. It's about that it would rather make L-malic acid than it would make D-malic acid. And it also uses milder reaction conditions. So I can work at really low temperatures. I don't have to work at these in the 1,000 degrees Celsius and high pressures. But then the downside of this is it again still uses fumaric acid, which is a petroleum-derived substrate. And also this enzyme is really expensive to buy as a pure enzyme, and it is also very sensitive to temperature. So if you do raise the temperature a little bit too high, that enzyme will not work anymore. So now I've got this whole range of things. I'm being really demanding, you know, being the millennial that I am. I want all of the good things without the bad things. I want low cost, I want higher selectivity, I want this and this and this. So then, how am I going to get this? So I get microbial fermentation. So that means that we actually use microorganisms inside a bioreactor. We give them a nice little home, give them all the food that they want. We hold them hostage so that they can make all the things we want them to make. So the way this works better is that there's a wide variety of microorganisms that can actually achieve this for us. They also have higher stereoselectivity. They also have higher productivity and, again, lower temperatures, those milder reaction conditions. But now, which organism do we choose? There are so many to choose from. So ideally, I would make an app. Anyone listening to an idea, I would call it like BioLinder. So it's like biological Tinder. And I would like make this list of characteristics that I'm looking for in my microorganism. You know, I want something that's non-pathogenic. I want it to make only malic acid. I want it to use a variety of substrates. I want it to work at any pH I want. And then I would just like swipe left and right to whichever fungus I want. But, you know, it being research and me not really having a say, I showed up and here was this guy, Aspergillus oryzae, waiting for me. And since it's so long to say Aspergillus oryzae, I have just dubbed him Perry. And this is not the same Perry that Professor Grimm made us lug around. This one treats me way better. So, what can I tell you about Perry? Well, Perry is a really nice green color when you grow him. He's aerobic, meaning he uses oxygen, and he is non-pathogenic, so he doesn't cause diseases. And the chemicals and the acids that he makes, it's not toxic to us. And then also, Perry is known as a filamentous fungi. So fungi, when I say fungi, you immediately assume mushrooms. So that's the same family. And filamentous means that if I put Perry in this reactor, he will make these tentacles and he will just reach in and get all those yummy nutrients that he wants inside there. And also, Perry has been used in the food industry for more than 2,000 years. So that sake that you like to drink there at, you know, Kung Fu Kitchen, or that soy sauce or that miso, that's all made from the fermentation using Aspergillus oryzae. And also, those enzymes that we use in baking and brewing, also Aspergillus oryzae. So now that we have decided what we're going to make and what we're going to use, the question now is, how do I actually ferment Perry? So it's called the recipe for my recipe for malic acid fermentation. So traditionally we use a two-stage fermentation that consists of a seed and acid production culture. So what this means is I would take a flask and I would grow Perry now so he gets nice and strong. So that's where the 4th years are now. Your parents have made you nice and strong, and now you need to go out of the flask, you need to leave and go produce things, make a contribution to society. They're done taking care of you now. There's no more free food, now you've got to work for it. Then we've also got a really high glucose concentration. So the nice thing about Perry is we don't have to worry about like diabetes. He likes sugar. We can give him lots of sugar and he'll eat it and make whatever we want him to make. Then we also need to give him a limiting amount of nitrogen. So that means that in order for him to make malic acid, we want to limit the nitrogen that he has, and that will actually shock him into making malic acid, or so the literature says. Then we need some inorganic salts. So these are just things like phosphates and chlorides and sulfides, you know, all those things that we get to do normal basic functions inside our cells. And then, since this is a fermentation where we're making acids, we need a neutralizing agent, which means we need something to control the pH. It gets really acidic in that flask. So if I'm not bringing the pH up, Perry will not be very happy with me and he'll just die. So now that I've done all of this, now I have my recipe, I can start with my fermentation. But since engineering and the word lazy are quite synonymous with each other, I decided I want a one-stage fermentation. Since we are working with biological systems, I don't know how many people know this, but you have to work really sterile. Just Perry is the same as us. He doesn't want some random microorganism coming into his house and eating his food and, you know, pushing him out of the way. So we need to work really sterile. So the one way we can do that is by having one culture. So now Perry will grow and he'll produce acids in the same culture instead of us having to displace him into a new broth. And then we can see a picture of Perry. So we see those filaments that I was talking about where he reaches in and gets the nutrients. So now we have one, but now we still need a neutralizing agent. So what will we use? Well, we use calcium carbonate. It's this nice white powder. So it doesn't cost a lot. It's quite a cheap material. It's abundantly available. And then the nice thing about calcium carbonate is it doesn't readily dissolve. It has a really low solubility. So you get this white sludge in there. And as the pH keeps going down, the calcium carbonate will dissolve and increase the pH again. So it's this system that works on its own equilibrium. So I don't have to stand there and check on it the whole time. I'll just add it in and it does what it needs to do. So now the question we have to ask ourselves is, how much calcium carbonate do I add? So if I was really nice, I would go every day and give him the little bit that he needs. But I just want to know, if I add everything in at once, how will that affect him? So I added 3 different concentrations. And here we can see them. So I added 20 grams per liter calcium carbonate, 80 grams per liter calcium carbonate, and 120 grams per liter calcium carbonate. So we can see in the first flask that most of the calcium carbonate is actually dissolved now. So that means that we've used up that buffering capacity, and the pH will be much lower than in the other flask. So Perry will now stop making acids for us, because he's like, no, this is an inhospitable environment. Even though he brought it upon himself, he will now cease to do what we want him to do. Then the next one, we have a little bit more calcium carbonate still stuck on the side. So since we're shaking this flask nice and slow, you know, getting him lots of aeration, making sure he gets everything he needs, he tends to grow on the side. So he has access to some of that calcium carbonate, but then leaves nothing for the rest of them here at the bottom. So once again, we'll find a drop in pH, but not as much as the first flask. Then the third flask, we can see I've still got lots of calcium carbonate. So this fermentation can still keep going. So now I know that if I look at the 3 flasks, I can see that there is a difference. But now I need to know what is actually happening inside. Like, is it different or is it pretty much the same? So we used a black box model. So what a black box model is I have a black box, I'm putting in certain things and I'm expecting an output from them. So we are giving Perry glucose, he has access to oxygen, and we give him some nitrogen so he can grow. So what happens? Perry grows and then he makes everything. He's like a woman, he doesn't know what he wants to do. So he just makes everything. So I measured everything we could possibly measure on our equipment here, and then I've listed them there. And then he also makes some CO2. So why that's important, we'll also see. So now, how do I solve the system to understand what's happening inside this flask? So I did a carbon balance, I did a nitrogen balance, and I did a degree of reduction. So I've got 3 specifications. Then I measured all of those things, metabolites, on a very fancy machine called high performance liquid chromatography. So you put it into a little vial, and this fancy robot arm like takes it and takes a sample and like tells you what's in there. So not a lot of work, which is really nice. And then we have these nitrogen tests. So these are little vials, we put samples in, and then it tells us the nitrogen content. So now I've got 12 things in my model. I have 3 specifications and I've measured 9 things. So now I've got a fully specified system. So now I can understand what is happening inside the flask. So what I did is I used a carbon distribution. So I said, if I give him this amount of carbon, how will Perry distribute that? How will he decide what do I make? How much will I send this way? How much will I send that way? So I took how much he made and I normalized that to 100%. And I said, everything that's present here is 100%, and I broke that up into the components. And then I got this graph. So at the bottom, we can see the components, and then I've got 3 columns. That's the 20 grams per liter, the 80 grams per liter, and the 120 grams per liter calcium carbonate. So the first thing that we notice is that with the first flask, Perry ate some, and then he was like, no, I'm full now, I'm done, it's okay. You know, I've made a little bit of this, so my day is done. Then in the second flask, he ate a little bit more. So here he was at home, And here he went to visit grandma, so now he had to eat a little bit more, you know. And then in the third flask, we had complete glucose consumption. So even though we still had so much calcium carbonate that was not dissolved, we didn't have enough glucose for him to continue, so he ended up starving. I'm really sorry about that. Then we get to CO2, which is one of the really interesting ones. So what happens with malic acid inside Perry is that when he takes glucose and he goes all the way down these really intricate reactions, and we get all the way to the tricarboxylic acid, what we call the TCA cycle, for him to make malic acid, he actually needs to fixate carbon dioxide, which is really cool because now we have a carbon negative process. So all those carbon dioxide we were sending to the birds, Perry can now take it and make something from it. So what we're actually seeing is that when calcium carbonate dissolves to control the pH, it breaks up into calcium and it produces CO2. So what Perry actually does is he fixates that CO2. So now instead of having to use his own sugars to make carbon dioxide, he's actually getting it from the calcium carbonate dissolution. And we can see this huge jump in malic acid. So from 20 grams per liter to 80 grams per liter, Perry made 3 times more malic acid. And then from 20 to 120, he made 4 times more malic acid. So now we can see that, you know, adding calcium carbonate, he likes that. He wants more of that. So, and then if we compare the minor components, those are now the ones that together make a very small difference, such as the biomass and all those other small acids that we also measured. So now we've got to ask ourselves, well, I forgot to do that one. Sorry, the increase. So what are our main findings? So our main finding was the improved glucose consumption. So he will actually carry on consuming glucose with more calcium carbonate. We've got improved selectivity towards malic acid. So again, he would rather make malic acid than anything else, which is what we want. We want him to make up his mind and rather just make malic acid instead of making all the other things. And then another thing that we found with the nitrogen testing is that we have incomplete nitrogen consumption, which means that when we put Perry in the flask, he is not going in there and eating up all his food all at once. He realizes, oh, you know, I've got a little bit of food, so I need to like starve myself little bit, and not eat everything all at once. So this suggests that nitrogen starvation is not necessarily the key to malic acid production. So then what is the key to malic acid production? Well, we can deduce it's not nitrogen limitation, and we can also understand that by adding calcium carbonate, we're not just acting as a neutralizing agent, but we're also adding substrate, we're adding carbon dioxide. And so why does Perry make malic acid? I don't know. So I can tell you when he will make more or when he will make less, but unfortunately why he makes malic acid is still a question I'm trying to answer. Thank you.
Our last speaker is almost finished with his PhD, and I'm honored to join them.
Good evening, everyone. It's a pleasure to welcome you to our presentation evening. I'll be discussing with you tonight some research in our group. I've titled this Revisiting the Theory of Combustion. This was under the supervision of Johan Labuschagne and Mike Heidenrich. They've helped me a lot in understanding some of this, and I've also helped them understand a little bit. Please pay attention because there will be a test after my presentation, so take notes, all right? I'm focusing on the theory of combustion as it relates to the development of biofuels. In our research group, we focus on the catalytic pyrolysis. So we take woody biomass, specifically some wood, and we put it into our reactor and we make pyrolysis oil, about 60% on a mass basis. Combustion is very important. It's existed since the dawn of humanity. It's assisted us in frying our chicken, in drying our meat, in burning our witches, warlocks, as well as our toast. All right? if you go around the globe, you'll notice that biomass is very abundant. And so for centuries, we've been using biomass as the main source of carbon and hydrogen. And then we burn it and we release the energy. Biomass actually specifically mines carbon from the atmosphere for us, and it uses solar energy to do this. It absorbs solar energy, it makes sugar molecules, and the sugar molecules are polymerized into the most abundant polymer in the world, cellulose. Biomass also utilizes water, and it releases all the oxygen, which allows us to breathe, thank goodness for that. It's a very, very important bit of information that biomass uses water and carbon and releases oxygen, because the water cycle is as important in the hydrology of the globe. What's very interesting is the trees, the vascular plants, etc., take up a lot of water, but they only utilize about 5%. 95% is actually released back into the atmosphere, and all the condensation that the biomass causes also ends up as a pressure gradient, a low pressure over the continents, which helps to draw in moisture into the continents, and so sustains biomass production. And so this is a sustainable mining of carbon from the atmosphere. Carbon is my main resource, but for combustion we actually need a whole lot of things. We need some fuel makeup, made up of carbon, hydrogen, the energy that comes from the sun, as well as oxygen to do the oxidation of our fuel. When we want to develop a biofuel, we have to ask the question, what do we want? And mainly for fuels, we're looking at energy quality. How much energy can I get out of the fuel when I combust it? Obviously, I don't want to burn a kilogram of X and get out a very, very small bit of energy, and I have to now maybe hop on my bicycle and do the rest of the work myself. If we look at the fuel, for example, fuel can contain carbon as the atom. If we burn carbon, it will release, for every kilogram burnt, 33 megajoules of energy. For hydrogen, hydrogen releases a lot more energy, specifically 142 megajoules for every kilogram of hydrogen that we burn. If we mix hydrogen and carbon together within the fuel, then we can also make our octane, which we can run cars off of, our diesels, methane gas, if you want biogas production, etc. You can see that adding carbon to hydrogen dilutes the energy quality of hydrogen from 142 to 56 to 47, etc. The more carbon you add, the lower your heating value. If you also add oxygen to your fuel, so let's talk about sugar, for example, C6H12O6, then we've already partially oxidized the fuel because there's some oxygen there already, and you can see that for sugar we have a low heat of combustion, 16 megajoules for every kilogram we burn. And in my case, the fuel that we work with, pyrolysis oil, wood is a precursor, wood contains sugar, so pyrolysis oil also has more or less the same heating value as our sugar. We just remove a little bit of oxygen from the pyrolysis oil and we end up with a slightly higher energy content. In my research, we throw some catalysts at the process. We've heard what catalysts can do. Mine try to remove the oxygen from the fuel, and so I can try to go up the scale to make my fuel release as much energy as possible using the least amount of mass. So, we know that some fuels are hotter than others, and some fuels give us more energy out than others, but we don't exactly know necessarily why. We measure heat of formation, but heats of formation are obtained from heats of combustion. And we use heat of formation in order to calculate heats of combustion. So it's sort of circular reasoning. So this doesn't tell us much information. And we were trying to publish some of our research in terms of showing that oxygen has detrimental effects to fuel development, fuel physical properties, etc. And we came up with some very cute correlations where we correlated the heat of combustion with the oxygen required for combustion, but the peer review process asked us, why? Why is your correlation much better than the rest? And so this forced us into looking at the thermodynamic fundamentals. If you go to the literature, for the last 100 years, from the late 1800s, early 1900s, the correlations for the higher heating value, delta H over here, is correlated with respect to the elements contained within the fuel: hydrogen, oxygen, carbon, nitrogen, phosphorus, chlorine, etc. And we put lovely fudge factors to make everything fit together. Okay, so this also doesn't necessarily tell us why some fuels are better in quality than others. So what did I do? I went back to the fundamentals. If I have a system, matter has a lot of bonds. When I combust the matter with one another, the bonds change, etc. I can look at the specific bonds for the fuel and the bonds for the oxygen, and I can see, okay, I require 432 megajoules for every mole of hydrogen bond broken, as well as 494 megajoules for every oxygen bond broken. The oxygen bond, just as a side note, is a very weak bond to break. So it's very nice to actually use oxygen as the oxidant. You don't have to put in a lot of energy because it's a double bond, so you're breaking two bonds at once. Now what happens in combustion? You reconfigure your atoms, and then they reconstitute as your combustion products. And the hydrogen-oxygen bond has a value of 459 megajoules for every mole of bond that I need to break or combine. Now, if I do some maths, I say on the left-hand side, this is how much energy I need to put into the system. On the right-hand side, minus 459 megajoules, this is what I get from my system. And if I add all of that up, I get the lower heat of combustion for hydrogen. And this value is calculated in the yellow, and it is approximately the same as the value that we actually measure in the lab. Okay, so there's a slight difference. I think this is 6% out. If I repeat this and I look at all the other ways that I can calculate this bond changes, I can decide to say, well, what is the specific change for hydrogen? Hydrogen has a bond within itself, hydrogen-hydrogen or hydrogen-carbon. How is the energy changing with respect to hydrogen from its initial phase to its final phase? And I can do the same for oxygen. Oxygen changes from the initial phase to the final phase as oxygen to water or oxygen to carbon dioxide. And if I add up these numbers, I more or less get the same value, right? So if I repeat this over and over again, I can see that I can come up with an equation. This equation looks very hairy and scary, and so I will give you a formula sheet with your test just now. All right? But what this equation says is that the high heat of combustion is simply equal to the change in bond dissociation energies with respect to your fuel, plus the change in bond dissociation energies with respect to the oxidant, in this case oxygen, plus other bond considerations like hydrogen bonding, resonance energies, etc. for benzene. And this equation can then almost fully describe all the bond characteristics and give you quite an accurate measurement. So what did we do? We wanted to apply this equation to more and more species, lots of different fuels. We had a database of 1,000. I gave up at about 359 because I had to count every single type of bond within each fuel. And this took months, so very, very long. But if I plotted the data and I wanted to see what change in carbon happens in terms of its energy changes going from the fuel to the combustion products, I see that hydrogen contributes 0 or negatively to the higher heat of combustion for fuel. This means that carbon actually takes up some of your heat of combustion, based on the calculations. With respect to oxygen, we see that oxygen actually contributes 100% based on its change that it undergoes. So this is between 100 and 200%. So you can see oxygen is quite a significant contributor to the release of energy during the combustion reaction. So if you look at hydrogen, for example, and nitrogen and whatever else is contained, you'll also see that there's some mild contributions with respect to hydrogen towards the higher heat of combustion. Hydrogen only adds a little bit of energy as it changes from its one state to the other state. So if I look at this equation, it's very nice, I can do a lot with it, except if you give me coal and wood. These are very complex materials. I cannot fully specify all the bonds in this piece of wood. It's too complicated. So this equation is completely useless. So what I actually did is I looked in more detail at this equation, and I decided to see, well, my heat of combustion, how does that relate to the change of bond association, dissociation energies for fuel and for oxygen? And so in the graph you can see there's a linear correlation between the change of bond association energy with respect to oxygen, and there is no change more or less for the fuel, while the heat of combustion is changing. So there is no functionality with respect to the fuel. So this is very nice. Oxygen has a strong, or the high heat of combustion is a strong function of oxygen and not a function of the bond association energy changes with respect to the fuel. And so I can cross out some term in the equation with respect to the fuel. Okay, so I've simplified this equation, but I'm lazy, right? I want a bit more simplification. So I have a look again at the term in the green, which is the bond association energy for oxygen. And now I look, how much oxygen do I require for combustion, for complete combustion to CO2 and water, the combustion products, and I plot it against the change in bond association energies. And I see, oh, there's a nice relationship again. So I can use this relationship and come up with a modifier function, all right, which I've specified there on the right-hand side, mu dh. All right, then I plug this in and I'm still not satisfied. My laziness knows no bounds. So I do another graph, another plot of some data, and now in the purple I've got the mass fraction of fuel required for combustion from 0 to 1, and for oxygen the mass fraction required for combustion. And where these two intersect, where the mass fraction of fuel is equal to the mass fraction of oxygen, I get some nice symmetry there. It's equal to a higher heating value of minus 13.87. which is also the constant that I came up with in the first place within my modifier function. And so I can actually approximate the top equation with a more simple equation shown over here. The last little bit over there is actually just a magic trick that I performed. I can just rearrange it quickly and just state it as the mass of oxygen required for combustion for every 1 kilogram of fuel I use for the combustion process. My simplified equation. Thank you very much. Now, this is a very beautiful equation because it's a one-parameter equation describing the heat of combustion as equal to some constant times by the mass of oxygen that I require for combustion. And this is what we were asked to do, and we were able to publish a paper on this and share our lovely correlation with the world. We also validated it against all the other correlations that you can get in literature. There are 24 major ones from 1900s, Dulong's equation, right up till 2000 and odd years. With a universal correlation. This correlation is inclusive for hydrogen, carbon, nitrogen, oxygen, phosphorus, sulfur, etc. So it really encompasses quite a varied, complex fuel composition. We used 1,087 data points to validate the correlation statistically compared to a lot of other correlations which I have not shown here. The top performing one in the literature was Boy. And you can see that our correlations statistically perform very well. We weren't going to look at trying to make an accurate equation. This was actually just to show the relationship of oxygen and its contribution to the heat of combustion. We also just have very nice straight graphs. You can come and talk to me later if you want to find out a bit more. But this just shows the linearity between our modified equations against the measured data. Now, in terms of the research that we do with regards to biofuel development, right, these have very important implications for us. Firstly, it tells us that we must reduce the amount of oxygen within the fuel, because the more oxygen the fuel contains, the less potential it has to oxidize further, obviously. We should reduce oxygen in the form of CO2. So when we remove oxygen from a wood-based fuel, we want to actually produce CO2. We want to avoid the production of water. So oxygen must not leave the fuel as water, because hydrogen does contribute mildly to the heat of combustion. You saw it's got a very high heating value. So we want as much hydrogen to stay in our fuel as possible, and maybe sacrifice more of the carbon instead. And that concludes my presentation. Thanks very much.
Well done. So what remains is the prizes. I call Dr. Nelson Safara. He's from the SAPI Tech Center. Is he in the audience? And Prof. Tafal, they'll be giving the prizes. You can just stand over here. Our first prize is the presentation we had earlier tonight. Our best oral presentation for the undergrad goes to Lesego Siabi. And then our next prize goes to the best undergraduate poster. That will be going to Mia van Skalkwyk. And then the best overall project, that's for the undergrads, go to Darren Ruth. Then we have our free oral presenters from the postgraduate group. They can all come together, Bianca, Monique, and Ryan. And lastly, we have the best poster from the postgraduates. That goes to Voldulexo. So I'll be handing over to the head of department, and then we can have some food.
Right, I see that we are seriously, into reserved time or special time. So all that I need to do is to explain to you why I asked Dr. Safara to hand over the prizes. If you look at the back of the brochure, you will notice that we have a variety of sponsors. SAPI has been a sponsor of this event for many years, and they are also, well, we are a little bit quiet in terms of how much every sponsor sponsors, but you will understand that the reason why I asked Dr. Safara was obvious. They sponsor the posters, the whole poster event. That's why all the posters have the logo, and thank you very much for that. But I also have to mention that all the other companies, we are extremely thankful for your contribution. Without sponsorship, we simply cannot do this. So just before we depart to enjoy some food outside and to have a good look at these posters, one of the posters is called Science Friction. And I mentioned to somebody outside that when I was a first year student, we did chemical practicals, and we had to analyze for all sorts of things and hand the document in for marking in a box. So you dropped it into the box, and then some clever student put up a little sign that said Science Fiction Postbox. So if you have a look at the posters, you might discover some science fiction. I need to thank the sponsors I've done. The academic staff I can only thank once more. It's your own fault that you are here. We claim that we want to be teachers, that we want to be academics, and if you start making calculations, you'll realize that the effort that we put in never equalizes the results that we get. So it's always a balance that doesn't work out. But I have to mention Professor Philip Kraus. I've got to mention Professor Willie Nicol, and I've got to mention Elisabeth de Tooy. In the same way as the undergrads were forced to meet deadlines until they produced what we are enjoying tonight, Willie was doing the same in a much more democratic way. So he passed it on to the postgraduate students, but it was a screening process that took a very long time. So to the three of you, thank you very, very much for your special contribution to this. I have to mention Don LaRue, who is sitting on that side. Don is the external examiner for the undergraduate CSC 421 module. He read through every single report, article, judged the posters. So it's a long and tedious process. Don, thank you very much again for your effort. But then I need to ask Gerry Klaassen, Andrew Macy, Kenny, and Paul Sonnendekker just to quickly come here, if you are still in the audience. Can I hand this to you? Gerry is the guy who did a lot of the work behind the scenes. So, Gerry, thank you very much. All the poster boards, all the organizing that had happened in this area was Gerry's responsibility. Gerry, thank you very much for that.
Thank you very much.
And just to mention why I have these two other parcels here, Paul Sonnendekker is a guy with a very straight eye. So he is responsible for putting up the posters so that you don't see any non-levelness. So Paul, thank you very much for that. And then lastly, but very important, we were without the secretary in our department for the best part of the previous year. And on the 1st of September, we had a new secretary, Jamaisi Kenny, and I think she fell into the deep side. So I wanted to thank her, especially because she did just about everything else. So thank you very much for them. And from this point onward, I would just like you to enjoy the evening. So please have some snacks, have something to eat and to drink, and study the posters. The test, Willie will walk around and do his little IQ test. Thank you very much.